I am here.
I guess we can start. I don't want to steal your presentation time, so stage is yours.
Thank you so much, Engin. So let me share my screen. Okay, so can you see my screen?
We are waiting. Yes, right now we can see it.
Okay, so is it full screen?
Yeah,
All good?
it's full screen.
Okay.
All good.
All right. Thank you so much. Thank you so much for the kind introduction and the invitation to present at ISCB RSG-Türkiye webinar series. As mentioned, and I just want to quickly also mention that I am actually currently the president of EpiBioNet, so it's a great pleasure to be able to also speak here on behalf of EpiBioNet. Although I'm not representing EpiBioNet right now, but as the president, it's a great pleasure to speak at this RSG because EpiBioNet is one of the affiliates of ISCB. So it's really nice to be part of this activity of the RSG. Okay, so I know it's pretty late in the evening. People might want to rush home and or might be on the way home. So let's get started. So the title, as you can see, it's going to be all about viral dynamics from the perspective of what we have been doing. But before that, I'll just give you a quick introduction. Okay, so let me hide this. Oops, what happened? Okay, so well, I think many of you may have seen this slide given the current corona pandemic. So we are currently in the midst of a pandemic. So when it comes to viruses, we are very familiar these days. But this slide is meant to remind us that corona is not unique. This COVID-19 pandemic is not unique. We actually have been afflicted with a number of viral pandemics in the course of human history. These are some of the major ones. Of course, here we also have other pandemics that are non-viral origin, but we are going to focus on the viruses. So we have the so-called, so we have smallpox, we have yellow fever, we have HIV, of course, a big one. Spanish flu was a big one. We will be actually talking about flu later, so this is quite relevant. It's estimated that it took the lives of 40 to 50 million. Then there was Russian flu, a small ball there. Asian flu, Hong Kong flu. So there's a lot of flu episodes that keep recurring. Of course, every life matters. And of course, if we take from the perspective of every life matters, these numbers are really huge. And then we have SARS, Ebola, MERS, swine flu. So of late, we have been having a lot of viral pandemics. COVID is the current one. The numbers I checked today, we have, I think, more than 2.4 million, if I'm not wrong, number of deaths. So that's huge, a big loss to humanity. So yeah, so there's a lot that's being afflicted by viruses. So it's very important that we have a good understanding of their diversity, because one of the big challenge with viruses is that they keep changing. Even in the light of having a vaccine that is being rolled out, a constant fear is what if the new variants evade or not effective against the existing vaccine that we have. So today, hopefully, we will, the take-home messages will be, something. Hopefully, you will understand or learn something about viral changes, viral diversity. Okay, so this is just to remind us. Although there are many viruses out there, these are some of the most lethal ones that have been listed by WHO. So Ebola comes under filovirus. Then we have Marburg virus, Lassa fever virus, MERS. You may have heard of. Zika, I think, has been around of late, so you may have heard of that. Henipaviral, maybe not so common. We don't hear much about it. Rift Valley fever virus, SARS, of course, the current one, and Crimean Congo happens to be actually quite predominant in Turkey. So these are some of the most lethal viruses, and it's good to be aware of these and keep a lookout. Now, viruses are actually something that I've been fascinated with. I've been very interested in studying viruses. One of the main reasons is because there's a big abundance of data available publicly. There's just so much data available for free. Anybody can access these data and study them. And also the fact that the genomes are rather small, compact, that also makes it easy to study, but not easy for the sake of just being easy, but rather I mean, they're so important. At the same time, you have this availability of data. The genomes are small, so you get the whole genome available. And for a lot of big organisms, whole genomes are not easy. Of course, they're becoming much easier these days, but for viruses, actually whole genomes have been around for a much longer period, given their small genome size. All right. So, for example, dengue virus, about 30,000 nucleotides. So, people have been doing full genome sequencing for quite some time now. So, you had actually access to the full genome, so you could understand, how the whole virus, works. So that's one of the key advantages of studying viruses. Data is in abundance. This is just to show you how rapidly the data grows. So, I mean, yes, it's an abundance, but how big are we talking about? What is the size of data? This is just to give you some insight towards that. This was actually data collected by one of my former students, Chong. So we actually were just tracking from September 18 to November 2019. And so for viral proteins, we went from 4.9 million to 5.8 million. So there was a growth of about 18%. And for nucleotides, there was a smaller growth, and we went from 2.8 million to 3.17 million. So where do we stand at the moment? Actually, the last I checked, sometime this year, we are more than 7 million now for viral protein sequences. All right, so yeah, they continue to grow, and over a period of a year, about 1 million. It's already a lot of sequences. So it's a big number, and it's also growing rapidly, which also makes it interesting to do comparative analysis. You could actually do time series analysis just to see, how evolution is impacting, or how they're evolving, how they're changing over time. So there's an additional benefit of having this big availability of data that's increasing rapidly. So where exactly do you find the data for viruses? So of course, NCBI, as everybody knows, it's the big resource, primary resource for these sequences. So you can go to NCBI, for example, the nucleotide or the protein database. However, I wouldn't recommend to go directly into NCBI protein database or nucleotide database. I would suggest to go to the taxonomy browser and then search for the species that you're interested in, and then from there it will bring you to the protein on the nucleotide, because as you see the table here, so if you click on the numbers, this will get you to the protein on the nucleotide, and then you get exactly what you're looking for, rather than the other way around. The other way around, you search by keyword, you do keyword search, you might not get everything, and you may have false positives. So it's much better to go through the taxonomy browser. And so you can search, and then whatever you search, you get this nice table under the direct links. You can click and you get the whole number. You get all the data available, which you can download through HTTP, FTP, or even API if it's too big. One of the nice advantages of the taxonomy browser is you see this lineage down here. So, for example, you might be searching for coronavirus and you get the number. You could actually click on any one of these levels, the ranks of lineage, and you can actually be presented with the data for just that rank level, which is amazing. Because, for example, if you wanted to study whole viruses, you just simply need to click on viruses, and they will give you 7 point some million sequences. You click the protein, and you can download, assuming you don't get timed out. Otherwise, you have to do the API approach to download the data. All right, so it's accessible, it's open, anybody can do this, and I hope some of you would explore this further. All right, so this is the lineage that I was talking about. You can really go around, play at any level of lineage you want. You can then quickly download the data and start studying them. So that's a primary resource that's accessible to anybody. NCBI also of late has been emphasizing, or has been putting an effort on this database called NCBI Virus, which is some sort of a derived database, or you could also, not really specialist, something borderline between derived and specialist database, or maybe in some sense specialist for viruses. What's nice is the numbers are actually pretty much matching. They might be a little up and down maybe due to annotations, but you could actually just come here and search for your viruses here. What's nice over here is that they actually have taken the metadata of the viral sequences and organized them in a very nice way. So you can, for example, have the geographical location separated as a separate column, the length, the host species, and a lot of other metadata that you get within the record. The GenPep record, for example, is nicely separated. So you get access to all that very easily. Otherwise, if you go to the primary database, NCBI protein, then from there, you have to yourself manually, split the metadata. And sometimes it can be quite tricky to do that. Unless, of course, if you're familiar with coding and all that, then there's no problem. So this database definitely makes it easier for people who are not so familiar with bioinformatics or do not know coding to get hold of the data already nicely separated, and then you can do your, all kinds of different analysis from this. So I also would highly recommend this one, and you could do, like build phylogenetic tree. There's a lot already that you can do on the fly, like align certain selected sequences or build a tree, right? So databases are becoming smart. I like to call them knowledge bases, KBs, and they are allowing on the fly analysis these days. Besides the NCBI resource, we also have specialist databases. These are in some sense the bona fide specialist database. Usually they're much more smaller, they're much more focused on a specific lineage of a virus. In this case, for example, Coronaviridae is a database. Actually, VIPR is also, it's somewhat like NCBI virus. It's focusing on some family or genus of viruses. And however, this database has a lot of duplicates, but it's a good place to start off. Also, you can also consider this. But similar to this, this is actually mimicking FluDB, which is for flu viruses. So that's very specific to flu virus. It has it allows you to download sequences, protein structures, do all kinds of different metadata separation. Which is nicely organized according to geography, time, or subtypes of the viruses. So these, in some sense, to me, these are specialist databases. So, a lot of the grouping or cleaning has been done for you. So you just go there, get the data, and then you can, you're much more ready to start doing analysis. Unlike if you go to NCBI primary resources, you have to do a lot more cleaning by yourself. And GIS8 is another good example, which has become actually a big resource for coronavirus. And GIS8 is also interesting. This is an example of a database where they kind of overtake the importance of the primary resource. So, for example, if somebody asked you, where should you go to download data for a particular virus, you would actually go to NCBI because everybody knows that's the biggest and the best one out there. However, for flu and now for corona, it's the other way around. I wouldn't even recommend you to go to NCBI. I would say go to GIS8 because they have a certain policy whereby they reward people who actually dump the data here, not reward monetarily, but in terms of acknowledgement and credit of the work that they're doing, which is not so much when you dump your data in NCBI virus. So that's why there's preference for this particular database. So you could go here and you will find that there's so much more data you will find here for coronavirus compared to NCBI. All right, so this has become the the main stop actually for data coronavirus sequences, and I think the same is true for flu. This actually started with flu and now for corona. So this might be the future of databases, specialist databases. That those that are very focused, they turn out to be doing a better job than the primary dump, databases, and I think it's a good thing for researchers because your data is very clean and you organize and you can get right away what you want, and then you can, it basically cuts down the number of cleaning that you have to do from your side. All right. So of course, and like GIS8 is also now providing this on-the-fly kind of analysis that you could do on the variation between the different sequences. So this is also nice. So the more specialist the database, the more quality analysis that can be done on layered on top of it. So this is one of the other advantages of specialist databases. So the take-home message is, if you want to study a virus and if there is a specialist database available for virus, you should explore that first, see how good it is, what can it do, what it cannot do, and then ask yourself whether you want to take the data from there or the primary databases like NCBI Virus or just NCBI protein database or nucleotide database. So that's one take-home message from here. All right, coming back to viruses, so we talked about the data. Well, another thing, of course, that makes viruses fascinating in terms of studying them is the fact that they mutate much more rapidly than, of course, eukaryotes, bacteria, even other viruses. So, for example, the RNA viruses mutate much more rapidly than the double-stranded DNA viruses, even the single-stranded DNA viruses. So this is, so we get to really see what's going on, in terms of the impact or effect of that mutation substitution, and how it has structural or functional change. So in terms of understanding how the substitution impact, the viruses provide an avenue for us to explore that, especially RNA viruses. So much so that, these RNA viruses, when they infect one person, it might be one or two, one parent virus, one founding virus. And then in few days' time, you have a few variants of these viruses in that one individual. And we term this as quasi-species. So how very quickly you start with one and you end up with a diversity of variants within one individual, so akin to a species, but quasi-species in this case. All right, so that's just highlighting the rapidity of the change that's being generated within a single virus, especially RNA viruses. All right, so with regards to this variation that I've been talking about, changes, let's quickly understand some of the terminology. So viruses change, the sequences change. Of course, by sequence, we refer to the genome. So your genome sequences change, and this is typically termed as genetic diversity. I think everybody is aware of this. All right, so now genetic diversity, these sequences that you see changing, I'm sure you are aware of synonymous and non-synonymous mutations. So some of these changes are synonymous, whereby they do not impact the amino acid that's being coded for. So there's no change in the amino acid, right? Unless you have non-synonymous mutation, then there's a change in the amino acid. So not all changes at the genetic level actually impact the protein or at the protein level. So protein diversity, you could say it's much smaller in terms of genetic diversity, although there's no direct comparison. But you can have a lot more genetic changes, but not all of them. So from that perspective, not all of them would impact the amino acid changes. So you have protein diversity. Now, so there's something else called antigenic diversity. Of course, when you hear the word antigenic, typically you'll think of antibodies, but in this case, I would like to define these changes that impact recognition by the immune system. And by recognition, I actually would refer to both T cell and B cell. So not just antibody, but even the T cell recognition. So what do we mean by antigenic changes? So you have these changes at the protein level. So your amino acid changes, but the amino acid changes, could impact the recognition by the immune system or may not impact recognition by the immune system. So not all changes may change or may abrogate or maybe improve the recognition by the immune system. All right. So those changes that affect the recognition by the immune system, for example, the T cell receptor, then those are the antigenic changes. So those are the changes that we like to refer as antigenic diversity, or the changes that impact recognition by the immune system. So naturally, this would be much smaller than the protein diversity. Again, you cannot compare directly, but in terms of the number of changes that actually impact the immune recognition is going to be much smaller. For some of those changes, maybe there's just no change at all, although there is amino acid change, but it doesn't really change at all the immune recognition. All right, so I hope that's clear, the difference between these. My work here, I'll be focusing more on the antigenic diversity changes. So I hope that helps. Now, with regards to antigenic diversity, it's actually, of course, very important to study this, because these antigenic diversity changes, can impact recognition by the immune system, which means you could have a variant coming in where the immune system is just not able to recognize them. So there's an immune escape, right? So if you have a vaccine, your immune system has been trained to recognize the virus, and then if a new variant comes, you are not able to recognize, so immune escape happens. And this is what prevents the buildup of specific long-term immunity against pathogens because of this change. So immune escape is one problem. The original antigenic sin. Now, I'm not sure whether you have heard of this, but this is actually something that was first discovered or first recognized with flu. And these days, it's actually a big problem for dengue. So it's a phenomena whereby you end up having immunopathogenesis, where the immune system ends up killing or actually impacting the cells of the host. So in the case of dengue, you, for example, could have an infection by dengue 1 and you build immunity. Then you have infection by dengue virus serotype 2, and your immune system, your memory cells are not able to recognize your memory cells actually recognize dengue virus serotype 2 and think of it as the same as the earlier one. So they trigger the mount a response, but that response doesn't seem to be effective because it's a different serotype. So they trigger the same response, but it's not effective. So you have this inflammation going on, but it doesn't seem to work against that that variant or that new virus from the serotype. So of course, the literature suggests that a new lymphocyte should have been triggered to recognize this as different, serotype, as a different variant, different infection, rather than calling the previous memory response. All right, so you could read more about this. Dengue is a big area where this is a problem, and this is one of the challenges for the vaccination efforts. So original antigenic sin is another area, and of course, in general, all this affect vaccine design efforts. So how do you overcome immune escape? How do you overcome original antigenic sin? Together, they pose a great challenge for those people who are involved in designing vaccines. One of the key things to note when you design a vaccine is that whatever vaccine you design should match with the circulating strains. So whatever strains that are circulating in the community, your vaccine should match so that it can protect you against those. But if the circulating strains change, then the vaccines it becomes problematic. So it may not be that effective. And that's why in the case of flu, you have annual reformulation where you got to, study what are the current circulating strains and then come up with a new vaccine that is in line with the current circulating strains, so you can recognize them effectively. You may have passed by clinics where they say, we have the new, flu vaccine available. So this is an annual problem that we have. So with the current corona vaccine initiative, we have to be fast. Otherwise, hopefully not. We don't want the viruses to have changed enough that the vaccines are not going to be very effective. So that's a big challenge. All right. So now, with regards to antigenic changes, I also want to just point and have the audience recognize that they are actually much more punctuated. By punctuated, I mean, here you have an example of a sequence, right? Okay, so let's say we generate overlapping k-mers. Now, let's say we choose the length of the k-mer as 9. Now, you may say, why 9? Well, I'm sure you're aware of the word epitope. So I'm not very familiar with the audience, so I hope you are aware of the word epitope. These are the fragments or segments of the viral sequence, let's say, in this case, viruses that are being recognized by the immune system T cells. So these parts of the virus peptides that are being recognized by the T cell receptor, these are the epitopes. So, and for epitopes, typically the length is 9. For class 1 is 9. For class 2, they are much longer, but the core is 9. So let's say we take 9 as a length for this example. So within this sequence, you could have 1 to 9 as a potential epitope, 2 to 10 can be a potential epitope, 3 to 10 is a potential, 3 to 11 is a potential epitope, and so on. So we have generated all the potential repertoire of epitopes that you could have. Now, if there is a mutation at, let's say, position 9, W changes to something else. Now that change in the W to something else can actually affects how many 1, 2, 3, 4, 5, 6, 7, 8, 9 can potentially affect 9 different epitopes that can potentially be recognized by the immune system. So it's punctuated in the sense that it has a much larger effect. So in some sense, just one mutation in the sequence can impact a region of 17 amino acids of the sequence, which which covers a repertoire of 9 potential 9 more epitopes. All right. So I hope that was not too difficult, but I hope you got the idea from this that these antigenic changes can have a larger effect. All right, so study of viral diversity. There are many studies of viral diversity. If you just search PubMed, you'll get a lot of papers. I wanted to just, of course, I'm going to be focusing on the work that I'm doing with regards to viral diversity. And so, of course, just like us, there are many people out there who do viral diversity. And in general, viral diversity studies can be divided into two. So you have the alignment dependent and the alignment independent. Alignment dependent obviously is the more common one, is the one that you see regularly. So you have your sequences, you got to align them. So you basically put the homologous regions in the same column so you can see what are the changes that are happening. All right, so that's alignment dependent, very common. That's what most people do, and what they do is they look for conserved regions, non-conserved regions, and we try to make some sense of the changes that we see. Now, with regards to alignment dependent, there's of course a big problem here. You will have difficulty aligning them if there's a big diversity. So if the viruses are very diverse, for example, if you take the envelope of HIV-1, it's very diverse, and aligning this protein sequence can be quite a challenge. Similarly, for influenza subtypes, you can take the major subtypes H5N1, H1N1. If you take the HA protein, the hemagglutinin on the neuraminidase protein, and if you try to align them, it can be quite a challenge. And of course, as you may be aware, multiple sequence alignment is a non-optimal approach. So you have to you use heuristic approaches. Whatever tool you use, it's going to be heuristic. All right, and if it's heuristic, you need to do eyeballing, you need to manually check the alignment, and you got to look for misalignments, and you need to fix the alignments. All right, so these are challenges when it comes to aligning sequences. So if they're very diverse, you got to be more careful. You got to spend some time honeymooning with the data and making sure that you have taken care of the alignment properly. Do not proceed to analysis unless you're very sure that you have done a good job there. In fact, I often joke that some people actually write papers and they use the word eyeball. We eyeballed the alignment for misalignments and then corrected them. Right, so there are certain methods to do that. We could discuss that if there's an interest there. But anyway, the main point I want to highlight is there's a limitation for alignment dependent. When your viruses are diverse, it just becomes difficult to align. Even if you do spend some time aligning them and doing the honeymoon, if it's really diverse, no matter how much time you spend, you just wouldn't know whether the alignment is correct or not. If it's just so many gaps and so many mismatches, there's no way for you to tell that this is a good alignment. So it can be very tricky. All right, so and this is studying maybe proteins from the same species. Imagine if you wanted to take different species from the same genera. Now, what if you wanted to go different species from the same family? So if you go above the lineage, from species to genus to family, as you go higher, you include different species. As you include different species, yes, they might be homologous. Well, they should be homologous because they're related. And if you take the envelope of one species, another species, another species, another species, and you align them, it's gonna break down eventually. The alignment will not. You will not be able to get global alignment for sure, unless they're really well conserved. In the case of flaviviruses, there's some good chance, but in the case of HIV, it might be almost impossible. So you have this problem. The alignment will break down as you go higher. If you go to a higher rank in the lineage. So how do we study viruses in such a case? How do we study across the family? How do we study across the genus? How do we study all viruses? Is it even possible to study the diversity of all viruses? There's no way you're going to come up with an alignment of proteins of all viruses. It's just going to be impossible. I can put my money there. All right, so this alignment dependent clearly has a limitation. It's very good, but it has that limitation. So we need alignment independent approaches, and this is, of course, it's not perfect too. It has its own problems, right? But there's also some approach there, and we have also done some work which I would like to share in a bit. In fact, right away. So alignment independent approach. So this is one of the work that we are doing, and we could talk about other things, but let me just quickly dive in the interest of time. Okay, so let me give you an example. So so the work that we are doing, we like to call this generation of a minimal set. So what do we mean by a minimal set? Let me give you an example. So let's say we have these 3 sequences. They are all unique, so they are not duplicates of each other. They're different. All right, so where is the difference? So you can see F and L. So at this position, 2 of them have F, 1 of them has L. At this position, you can see this one is Q, this one is L, and this one is Q. So taken together as a whole, these 3 sequences are different from each other due to 1 or more of these changes that you see from that consensus and variable residue here. So now that we know they're different, now let's generate, so I'm going to be focusing more on the antigenic diversity. So let's generate the repertoire of potential epitopes. All right, so I take the first sequence, and again, I'm going to use the k-mer length of 9 as the standard length of the potential epitope. So let's say I generate potential epitopes from each one of these. So these are all my potential epitopes from each one of them. Now, you will notice that some of these k-mers, or the 9-mers in this case, are exactly the same across the board. So those in bold here, 3 here, they're exactly the same. They're shared across the 3 sequences, okay? And then you see these ones in capital, but not bold. They're exactly the same with this 3rd sequence here. And then these ones here, okay, so these ones here are exactly the same between the 1st and the 2nd sequence, but different from the 3rd sequence. So what we see is that actually there are some parts that are shared across, there are some parts that are shared between 2 of them. So could we compress the number of sequences so that we have a minimal set that still captures the repertoire of the total data set? So that means actually we could say that the middle sequence in some sense is redundant, antigenically redundant. You could actually remove this sequence, the middle sequence, and you still will capture all the potential k-mers of this sequence. So for example, these ones are captured by this. This one is actually captured by both, and this one is captured by the 1st sequence. So we could actually reduce the number from 3 to 2. So that would be the minimal set for this data set of 3 sequences. All right, so in some sense, what we're trying to say is like, could we compress the diversity to just have that minimal set that captures the overall repertoire of k-mers that we are interested in studying? All right, so okay, so yeah, so this sequence is antigenically redundant. So we actually came up with an algorithm that does this automatically. You give it any data set, it will be able to generate the minimal data set for you. I'm not going to go into the details of this algorithm, but if there is an interest, we can talk about this. We actually just submitted a manuscript for this. So if it gets published, you'll get to see that. But yeah, so I'm going to skip the details, but essentially this summarized the concept behind this whole algorithm. All right, so so let me share with you an interesting result that we observed. So this is now for all viruses. So this was the data set as of 2018 when we had 2018 slightly later than 2018. So we had 4.9 million sequences. So we took all viral protein sequences at that time, about more than 4.9 million. So you see the number over there. And what we did first was remove duplicates. So we managed to actually cut down the number to 2.16 million. And so there was a duplication up to the level of 56%. So there's a big message or lesson here. So there's a lot of redundancy in these viral sequences that we have. All right. So for example, I said today we have more than 7.2 million or more. So you could roughly say 50% of that is going to be redundant duplicates, exact duplicates of each other, which you can remove and you don't even need to study them. Why do we have these duplicates? Well, there could be duplicate sequencing efforts. The viruses are in different countries, so different sequencing efforts. So for example, somebody in Singapore might be sequencing, Indonesia might be sequencing, the strains might be circulating, and even if they are not the same strains, think about it. You have one virus 1, virus 2, the mutation might be at a specific position. Let's say in protein 10 there's a mutation, which means if there are 10 proteins, so put protein 1 to 9 of virus strain 1, protein 1 to 9 of virus strain 2 would be exactly the same, because the only difference that we see is in protein 10. Even in protein 10, the only difference would be in 1 or 2 positions, not the whole protein. So if you think like that, you expect a lot of redundancy between the sequences. So even though the whole virus is unique, as a whole it's unique, but that uniqueness is very restricted, localized to few positions, and that renders many of the other proteins between them to be redundant. All right, so we were able to compress up to 56.7% and reduce that. So we ended up with 2.16 million unique sequences that were unique to each other. So we are, by the way, we are not referring to the k-mers. Of course, the methodology of how we got these numbers is by using the k-mer, but we are referring to actual records of sequences. So we were able to, cut down from 4.9 records of sequences to just 2.16 records of sequences. And then, of course, this one is just pure duplicates. Then we applied this minimal set approach using the k-mer, but applied that at the sequence level and removed all the sequences, as you saw earlier. We removed, so we actually removing actual sequence after we do the k-mer analysis, and we were able to reduce this to 1.3 million sequences. So this is a reduction of 16.7%, of course, relative to the 4.9 million, not relative to 2.16. Of course, relative to 2.16 is about 38%, if I'm not wrong, a reduction. But relative to the original, we managed to reduce it further to 1.32 million. So so what are some of the take-home messages? Well, there's a lot of redundancy. There's some real actual redundancy, about 56%, and there's about 16% of antigenic redundancy in our data set. So so for example, in that 7 point some million sequences, we probably only need to study, we haven't done this analysis with the 7 point some million, maybe the numbers increase to 2 million. So out of 7 million, maybe what you really need to study is just the 2 million of those sequences. So that, because they in some sense capture the antigenic repertoire of all the 7 million sequences, it cuts down the number of sequences that one needs to study and just focus on those. Of course, the benefit of this is you are in some way dealing with a big data problem. You have a big data of 7 million, you can avoid dealing with the 7 million and you just focus on those 1.3 million. So people without the computational resource to study that many sequences can, this is one way of cutting that down. So besides operational efficiency of reducing this redundancy, so you can just study much smaller number of sequences, there's some deeper insights with regards to this. So for example, do we expect as the data grows, do we expect the minimal set to grow rapidly? So for example, if we suddenly sequence a whole new bunch of viruses, then the minimal set is going to expand, because what we see is going to be novel and it will add up to the pool. But if we keep on sequencing related viruses, then the minimal set is not going to expand so much because you are already capturing the diversity of what's out there. So it's going to grow very slowly. Again, maybe whatever mutation you see may already be seen in the pool. So it's not going to be considered novel, right? So you have to really have very novel mutations or changes in order for it to be part of that minimal set. So either we discover a whole new family of viruses or species of virus. This pool is it's not going to grow much. Of course, it will grow slowly, but the more it grows, the more it grows, the less rapidly it's going to grow over time. I'm not sure if that was clear. It will grow, but the rapidity of that growth is going to get smaller and smaller, because as you grow larger, the chances of finding something novel actually reduces. Right, so we actually feel that it's probably going to hover around that, but it'll be interesting to do this analysis with the 7 million, now that we have 7 million, maybe 1 year later 8 million, to see whether that hypothesis is actually true or not. Maybe we do find some new species and all that, and it will change. Well, it'll be interesting to see this. So this was something in terms of diversity that is available, that is present within the sequences. Okay, so this is if you want to know more, you could. This is the paper. The unique mean is the tool that we submitted. We're hoping. We're waiting for the response to see how it goes, but we have a GitHub page available. If you're interested, you can look into this. You can get in touch with us, and we'll see how we can help you with this. All right, so the next one, I'm going to just quickly focus, move on to the alignment-dependent approach that there's a paper that we published last year. May last year, and we want to share the results of this work. So this is available. You can read about it. And there's actually there's a preceding work which was done on HIV. You can also look into that. So of course, with the flu work, we went a little further, but it's in some sense quite similar, and it also allows us to do some comparison between them. All right, so moving quickly. So I'm gonna not bore you with some details of flu. I'm sure everybody knows that flu is a major public health and worldwide problem. All right, so the genomic architecture, it's a segmented virus. So you have 8 segments, and in total they code for 12 different proteins. And the mechanisms of viral variability, the major ones are antigenic shift and antigenic drift. So antigenic drift is your mutation, antigenic shift is your reassortment, and to some extent, I think there's recombination, very minimal. So these are some of the forces of change that affect the forces that affect variability of influenza. There was actually a study done by Haney et al., a colleague of ours, long time ago, and she did this entropy plot where she compared the avian H5N1 viruses, and it showed the locations where there's more diversity and locations where there are less diversity. So this was good in the sense it told us how diverse the virus is. We wanted to actually go a little deeper to see if we could learn so much more than just beyond the panoramic overview of the landscape of diversity for this virus. All right, so this was the methodology. Same thing, we went and collected only H5N1, so it's a very focused H5N1 data. We collected both the human and the avian host reservoir data. Then the data went through processing, where we cleansed the data, removed irrelevant sequences, removed duplicates, and then we aligned the sequences, and we did the same thing. We did the entropy plot to see the panoramic overview, and this is where we tried to dissect the dynamics of sequence change from there onwards, and then we did a few other things which I'm going to cover later. All right, so the data was collected from FluDB. FluDB is a big resource. Of course, EpiFlu from GIS8 is another resource. I would recommend to go with EpiFlu. There was a time when FluDB and GIS8 were almost similar, but now GIS8 has taken over, so no point going to FluDB. Well, you could still compare, but I think EpiFlu will give you so much more data. All right, so I'm going to skip all this stuff. We did the usual QC of the alignment and all that stuff. So here's just an example of an alignment Okay, we can skip all that. So this is the data set that we had from different parts of the world. These are the 12 different proteins, and we had these are the numbers from the avian pool and from the human host. All right, so we had more data from the avian and much lesser from the human. All right, so with regards to the mapping the T-cell epitope antigenic diversity, which I mentioned earlier, what we did was we actually took the alignment and then we segmented as a Kamer. We took a Kamer window approach. So and of course the Kamer that we chose was 9. So 1 to 9, 2 to 10, 3 to 11, and so on. So for each one of those windows, we will then do the Shannon's entropy analysis. We will measure the entropy of that particular window, which will give us the overview, overall diversity of that particular window. Then we go to the next one, and we measure for each one of those. All right. So now, each one of these windows kind of represent your potential epitopes, and by epitopes, this picture nicely represents, excuse me, if this is your antigen presenting cell, this is your class 1 or class 2 MHC molecule. The antigenic fragment, or the so-called epitope, it binds to the MHC, and then this MHC peptide complex is presented at the cell surface where the T cells come. The T cell receptor then recognizes it. If it's foreign, if it's recognized, then an immune response is triggered. All right, and that's when you call it a T cell epitope. If there's no immune response being triggered, then it's not an epitope, because even self-peptides do get presented. So just binding to MHC is not enough. You have to have recognition by the TCR. So we are making an assumption that these Kamers are potential epitope. Actually, there's some explanation behind the assumption. If we have time, we can talk about that. Why do we assume every one of them as a potential epitope? We could discuss that. Yeah, in this sense, actually, for that window 1 to 9, we're assuming every one of them as a T cell epitope. We are not even doing prediction. We are not even checking wet lab data. We just assume they're epitopes. And there's a basis for that, and then we look at the diversity. We look at the diversity of that particular region. All right, and so we use Shannon's entropy to measure the diversity within that window of 1 to 9, 2 to 10, and etc. This is the equation. Some of you may be familiar with this. All right, so PIX here is the probability of a particular non-number I at non-number position X, and NX represents the total number of peptides observed at that position. Let me give you an example to explain this. So for example, I have an alignment of dengue virus 3, serotype NS2A is the protein of interest for this. And let's say I go to a specific position, 176. This is just the middle position. The actual position is 172 to 180. So that's your non-number. And the number of sequences you have over there is, let's say, 64. All right, so you have 64 sequences at that window, and from those 64, you find out how many different sequences you have. So you have 1, 2, 3, 4, 5, 6, 7 different sequences, and this is just the frequency of those sequences. If you add up, you will get 64. All right, so one of them is dominant, it's the highest one, 39%, and then you have those lower ones. So so NX here refers to the number of distinct peptides that you actually have here. So that's NX, and PIX here is the probability of each one of them. So basically, you feed the probability of each one of these into the equation, and you will get one overall number that describes the diversity of that position. So in this case, it's 2.40. All right, so entropy is affected by the number of unique peptides. So the number of peptides you have here is going to impact the entropy. The higher the number, the higher the entropy value, and of course the relative probabilities are going to impact. All right, so what's the lowest entropy we can have? The lowest entropy we can have is 0. What does 0 mean? 0 means you have just one sequence. Of course, you cannot have 0 sequence. You have one sequence here, but it's found 100%. It basically is the same peptide in all the sequences. So it's 100%, and you will have an entropy of 0. Entropy, in other words, is disorder. All right, so there's no disorder. Basically, there's just one sequence. It's totally conserved. All right, so now you go higher, you go above 0 as you have more peptides here. So you could have a lot of peptides here, but if this one is, let's say, 95%, and you have a lot of peptides within that extra, the additional 5% is represented by, let's say, by 20 of these, you will still have very low entropy because you have one peptide that's so dominant, up to 95%. So it's this interplay of the number of peptides and their relative frequencies or probabilities together give you this entropy value, which gives you an indication of the disorder at that particular position, and you have your entropy. All right, so entropy theoretically can be as high as 39. Of course, I talked about the 0 being the lowest, highest is 39. And so, of course, this is theoretical maxima. The actual maxima we do not know yet, but if we use HIV as an example, HIV clade B, not even the whole of HIV, because it's going to be difficult to align them if you go beyond. HIV clade B alone was already so difficult to align. We observed an entropy of 10. So entropy of 10 is just amazing because HIV is considered to be one of the most plastic, plastic viruses where it just changes so much. And we got an entropy of 10. So I hope you could use that as a benchmark of how diverse it is, as it gets closer to 10. All right, so we have not seen anything beyond 10 because we have not tried doing an alignment of HIV, all HIV. Maybe it's going to be higher. We have done alignments of some other viruses, which we will show you in a bit. So this was this is the result for dengue virus proteins. So, for example, you see the low troves here. So these are basically very conserved regions. If you see high number represents diverse regions. So this is how you read the plot. All right, so there's actually a tool available, ABK Ivana. We have a new version of this, Hunana, which we are able to provide, but we are still testing it out. So this is available. You can download this and you can do your own entropy analysis. This actually does a few other things. For example, mutual information analysis. So it's a great tool. So yeah, it's something recommended for you to explore. So we did the analysis, and so here are the results. Human viruses, avian viruses. This is the entropy value, the average entropy value. As you can see, there are some pockets where it reaches close to 8. So there are some parts, just H5N1 alone. Sorry, wait, the red one is total variance. Ignore that. Entropy is black. So no, the highest one seemed to be, I think, around 5. So that gives you an indication from here for something. So yeah, so the highest entropy you're getting is slightly above 5, or yeah, between 4 to 5, or slightly more than 5. There's certain pockets that go pretty high, but overall, as an average, individual protein average in black, it's actually pretty low. PB1F2 seems to be showing the highest. This is average for the whole protein, so not just the maxima at a specific location. So overall, it's not too bad. Now, what is this? The one in red, total variance. Total variance simply means, so let me just go back a little bit. So here, for example, if this sequence was a dominant one, total variance would be actually everybody else. So we actually label this sequence as an index. Everybody else is considered as variance. So total variance would be the frequency of everybody else besides the predominant sequence. And there's a basis for this. For example, if you use this sequence as a vaccine target, because it's the most conserved, in this case 39%, if you use this as a vaccine target, then the population is potentially exposed to everybody else as potential variants that they may be exposed to, with a frequency of 61%. So you have a 61% of probability where you could where the population could be exposed to variants. So of course, this is not a good target for vaccine because the frequency, the total variance incidence is very high. 61%, right? So we actually also did that analysis, total variance analysis, to see where are the positions where total variance is rather low. So those will be the positions that would be favorable for vaccine target selection. You want positions where the total variance is very low, which means the index or that predominant sequence is very high, and you have very little variance to deal with, and that's something is going to be favorable. All right, so proteome-wide, so this was individual protein average, proteome-wide average entropy of 0.8, total variance of 14%, entropy of 0.7. So there's not much difference between the avian and the human in terms of overall, disorder. So it's pretty low, which means there are many positions that are going to be of low total variance that one could select for vaccine design if they're interested in. Of course, this is overall. You still need to avoid those positions with very high entropy. Those positions need to be avoided, even though the overall proteome average is rather low. Okay, so the overall proteome average gives us some idea, but it doesn't give us the full picture. So you need to look at the actual plot to see where the positions that need to be avoided for vaccine target selection. But as a diversity, you could say, well, it's diverse as a proteome. It's not too diverse, but yeah, there are some proteins that are much more diverse than the others, and there are some pockets that are much more, disordered than the others. And we need to be aware, what are these proteins, where are these positions, so we can avoid them in terms of, vaccine design, for example. All right, so now compare this with HIV. So as you can see, the black again is the entropy, and the red is the total variance. The entropy of HIV, of course, it varies from protein to protein. For example, polymerase is much more conserved, so you see a lot of low pockets. Envelope, you see a lot of high pockets, and you can see here this is hitting more than 8. So some of the peaks are actually observed in HIV envelope and also NEF here. You see some close to 8 for rep. So these are so there's a basically a non-uniformity in terms of the diversity across the proteins and their positions that are much more diverse than the others. All right, so imagine these positions in envelope. They are very plastic. The mutation is just so high over there. It's just interesting why that particular position can mutate so much compared to some of the others. So and a lot of people have this thinking that HIV is diverse. Yes, it's actually diverse, but it's actually not diverse overall of flat diverse throughout. There are actually some positions that are very low. So you can see some in GAG, a lot of them in POL that seem to be quite conserved. So this is a revelation, and when we get this panoramic overview, we get to see what's going on across the board. So it's very interesting. We can learn a lot from these plots. Now, this is just another plot looking at entropy versus total variance, whether there is an actual relationship. So, of course, when entropy is very low, then your total variance is also very small. But as entropy grows, then the total variance, you see a spectrum. So, for example, at entropy of 2, you can have a total variance of 25% all the way to maybe around 50, 50 or 60%. All right, so this bulge that you see as entropy goes up. So in terms of vaccine design, I mean, you need to favor somewhere lower here where entropy is low, total variance is low. But as you go higher, close to 1, even at 1, you can see the total variance. There's a range over there. And we saw something similar for the human viruses. Of course, in our case, let's say we're interested in designing human vaccines, then the human data would be more relevant. But you see the same pattern in terms of the relationship between those. So for vaccine, one needs to really focus at the lower part over there. And this was for HIV. I think in the interest of time, I need to hurry up. So with regards to HIV, yes.
We have completed 1 hour. If you can wrap it up like in 10 minutes, because we're going to have questions as well.
Great, yes. All right, so so with regards to HIV, you see a slightly different pattern over there, but again, the lower part is much more compact there compared to these. So you don't see the same pattern across the viruses. So this is also an interesting analysis to be done to really see the relationship between entropy and total variance. All right. So the next up, we wanted to dig down deeper to see how actually they are changing. All right, so I'm gonna introduce you to some of the diversity motifs. So the index, you are aware, is the predominant sequence, and total variance is everybody else. We actually dissected or broke down the total variance further. So you have the next dominant sequence, or the dominant within the variance is called the major variant. So this is one single sequence that's predominant, and then you have the minor variance, where you have well, let me put it this way. So the major is the dominant within the total variance. Unique are those that you see once, and that's it. You only see them once in the alignment. So everybody in between is called minor variance. So I hope that makes it much easier. Nonotypes in this case, or chemotypes, is just how many distinct peptides you saw. So this would be one distinct. In the minor, you might have few distinct. And of course, in the unique, everybody is a distinct. So this is the number of distinct. So you have few diversity motifs. So the index, major variant, minor variant, and unique variants. And then, so this is just the definition. And here, for example, this position, there are that many sequences. The entropy is 0.6. This is the index sequence and is found in 92%. And so the variance is going to be 8%. Out of that 8%, major is 5%, minor is 2%, and unique is less than 1%. And this is just the percentage of nonotypes, not one distinct peptide, but the percentage from the data. All right, so we collated this data, and then we did a plot. All right, so let's look at the dynamics of the diversity motifs. So what is the pattern of the major variance and the index and the total variance? So so very quickly, the black line here is your index. So as your okay, so sorry, the red one is index. All right, so this is total variance, and this is motif incidence. So as your total variance grows, so at zero total variance, you have index 100%, and you saw that you see that the index is almost a straight line. That means in the whole avian viruses, we saw for each one of those incidences of index, we saw a data point, except for somewhere around here. So as the index dropped from 100%, so 100% is completely conserved, then you go to lower. As you go lower, of course, the total variance is going to go higher. So this crisscross is expected, but you can see the gaps where you don't see any incidences over there. So what happened to the major variant? The major variants, we saw they took a pyramidal shape. So now there's a reason why index. So the major variant cannot be bigger than the index. So, and of course, they cannot be bigger than the total variance. So by definition, they're kind of restricted by the total variance. And so they cannot they cannot go overboard the total. As you go down here, they cannot go overboard the index. So because of this restriction by the total variance and by the definition of index, they take a pyramidal shape. So we expect a pyramidal shape, but what we do not expect is where would they lie within that pyramid. So we get to see the pattern for avian viruses, and then we see the pattern for human viruses, and you can see there is some distinction between them. We see more, more of the human viruses tend to be on the lower side, whereas for the avian, they seem to be more spread out. So the major variants tend to be congregated here, and we can do an average for that. This one was the other motif. So for example, the unique, let's focus on the unique for now. Unique is unique is a motif where you see sequences that occur once. Now, you might think this could be sequencing error. They occur once and they disappear. They're never to be seen again. But the percentage of these unique variants was rather, high. They were actually there for every incidence of the total variance. So if they were just sequencing error, you wouldn't see them that much. You wouldn't see them throughout that spectrum. And also, you do see that there's a quite good percentage of them that are unique variants. So there is some plasticity in terms of, you see variants that appear and then disappear, never to be seen again in that data of the many years that we have collected. So that's unique variants. Of course, we expect them to be low, and the minor variants seem to be actually the main the principal variants across these, because there comes a point close to 80% when they actually overtake the major variants and they become the dominant one, despite being despite being of incidence smaller than the major variant. As a whole, they seem to be much larger than the major variant, when the diversity is, of course, much higher. When the diversity is around 60% or something, actually beyond 50%, we can start expecting them to overtake. And depending on the virus, we see different patterns. So, but in the case of flu, we see that they overtake slightly around 60%. In the case of avian, they seem to go slightly much earlier. But you can see there's some difference in the pattern. But the minor variant seems to be still the principal variant. All right, the total variance is just the distinct variance. So there seems to be. It's fascinating how for the human you see much more the range for the nonotypes of the human viruses. Now, if you compare this with HIV, which is again very interesting, you see the major variants are much more spread out. They've covered the whole range of everything that's possible. And for the HIV, the total variants actually go up to 98%. So you actually have an index of just 2%, and 98% is total variance. Imagine how many variants you will have to just occupy 98%. The dominant one is just 2%. And that's why we call HIV to be so plastic. And many of these are just unique variants. They occur once and they disappear. So there are some positions that are hotspots of mutation for HIV. And you can see the unique variants. And like here, they actually go above the major variant. So we actually have the unique variants overtaking the major variant as a whole, as a population. And just fascinating how, so we don't think these are going to be, these are sequencing error. And within HIV, we actually see that their positions where the unique variants are just so dominant. And these are the position, all these dots that you see here for HIV, we actually, we call them as hotspots of mutation, and they're very plastic, and they produce mutations that appear, disappear. Imagine you do a vaccine using those positions. Your next vaccine is not going to work because you will never see those variants again ever, well, at least in the data set that we have analyzed. And the nonotypes is, of course, much higher. All right, so that was the proteomic, and for individual proteins, we did the same thing, and we saw some variations. And this is again just a violin plot summarizing the same results earlier. That you see for the index, you see less incidence, much lower, and a lot of them at that higher level. Okay, so, and this was for HIV. You can see the index was they covered the whole range. You had index all the way from 100 to almost 2%. Index, as I mentioned, 2%, and the variance will be, total variance will be 98%. And so the minor variants are predominant there, and unique is also pretty strong in the nonotypes. All right, so, and of course, this is the same thing for the individual proteins. We also had something else which I may not have time to go about. There's something called index switching, which we observe initially for index, but later we observe for all the motifs. We call this now motif switching. So very quickly, in summary, there are positions at the borderline where something may be indexed at this, let's say position 1, it's an index. At position 2, of course, there'll be a new amino acid at 1 to 9, 2 to 10. So you introduce a new amino acid. What happens is the one that you expected to be indexed becomes major. The major kind of goes up and becomes an index. So this is a switch, index switch, but we can observe this with minors and a unique also. So motif switch seems to be very common, and this is actually another problem in terms of viral diversity dynamics. If you chose something as a vaccine target and it was the index, very good, but if it's possible that through mutations, it may not be indexed in the neighboring regions, and that could impact or affect the effectiveness of that vaccine, which we could discuss further. But this is another area, and we're actually building a tool called MOSWA by one of the students, and we are trying to come up with all the different ways one could study this, and hopefully we'll get that to the public community soon, so they could also look into the different ways switches are happening. All right, so I'm not going to have time for this. But yeah, this is just a crazy amount of switching that's going on from index going to index, some of them splitting. You also have mergers. And well, in summary, there's a lot of this index becoming major, major becoming index, or index going to minor, minor becoming index. So you have that kind of so-called crazy stuff that's happening among viruses. And of course, lastly, at the end of the day, for vaccine, you want to focus on those very highly conserved. So you want to avoid all this variability. So highly conserved, maybe more than 90% conserved, and you can do this analysis and you come up with those sequences that are highly conserved. Then you can look for epitopes among them and match with IADB to see if there's experimental data that tells that they are they've been shown to be immunogenic, and then you can take those further for preclinical trials and etc. So to wrap up, I hope in this rush I managed to give you some idea of how viruses changes and how you could study viruses. We're actually also working on a tool called WADA that will be able to give you all this. Not the switch is MOSWA. WADA will be able to give you this different motif, major, minor, and unique, and how the distribution looks for your own viral protein sequences. Hopefully we'll be able to bring that to you very soon. All right, so in conclusion, there's a large repertoire of viral variants in the proteome. Basically, there's a lot of diversity, and these variants influence the variability dynamics of the viral population, which we studied in the context of the different motifs. Each of the motifs has its own pattern, and together they represent the intrinsic patterns in the structure, function, organization of the virus that affects the quasi-species fitness selection of the virus to survive. We also have observed complex motif switching that affects the fitness of the virus. And of course, the good news is we do see highly conserved sequences that remain because they're important for function. These are the weak targets that should become the priority for vaccine design, drug design. So there's still hope in terms of overcoming the diversity of these viruses to prevent the inflection of human death and other morbidities. All right, so finally, I just want to thank all the individuals that have been involved in this various project of ours, and from NIH, Perdana University, Bezmialem, and even Tubitak. Thank you very much, and I will end it over here. Thank you, and apologies that I took the extra time. Back to you, Engin.
Thanks so much, Dr. Khan. I would like to thank you on behalf of RSG-Türkiye and on behalf of our attendees. Our attendees now can write their questions in the chat box. Just before sending it, please raise your hand so that we can know who you are. And I'd like to thank you again, because it was more than a webinar. Actually, it was almost an introductory course, so it was so good. And with your permission, we would like to definitely share this with our other followers on YouTube. That would be very nice.
No problem. Yeah,
and,
my pleasure.
I guess we have a question, but I guess there is a problem with the chat box. I will send the questions right now.
Wow. Okay. Singapore. All right, so let me just read it. Thanks a lot for this informative talk and accepting our invitation. As a RSG-Türkiye member, I'm joining from Singapore. I could not listen to the whole talk. Also, I'm not a virus researcher. However, I have a few questions. How do you decide very novel mutation, distinguishing novel from very novel? Yeah, and the first part, decreasing. Okay, let me answer that part first. I mean, that's a colloquial term that I used. By very novel, of course, what I meant was as the pool of this minimal set, let's say we have a minimal set of 1.2 million something, and then as the pool grows to capture novel sequences, novel k-mers, over time, it's going to be hard for you to have mutations that we have not seen before. Right, so mutations that we see may already be part of the pool. So by very novel, I meant, we got to see something. Yeah, in this case, maybe the definition of very novel would be if the pool is large enough, you might see a novel mutation, let's say, in that family of, maybe for that family it's novel, or for that species it's novel, but with respect to the pool of all viruses, maybe that chimer has been seen in some other virus. So it's not really a novel mutation with respect to the whole overall pool, but maybe with respect to that species is novel. So that's so in my definition of very novel here, maybe that's not the right word. Basically, something different from that pool of chimers, which are already capturing the diversity of so-called 5 million sequences. So for you to have something that to be included into that pool, it got to be different from that pool, and it becomes harder and harder. As this keeps growing, it'll be much harder for a mutation to be included as part of that, and it will only be included if it's going to be different from what's already there. So, and of course, mutation is random. Most of the times, whatever mutation we see may already be part of the pool, so they will not be considered, they will not be included unless we have a mutation that is very novel, different from that pool. Then the pool will expand in size. All right. So there might come a point when you can have all kinds of mutations, but they are just not novel enough. Well, first of all, they wouldn't even be novel with respect to this. I hope that kind of answered. Sorry to use that colloquial term, but I hope that gave you something. All right, so next question. All right, so decreasing 4 million to 1 million without a reference. I hope I have answered. Okay, so maybe we need to have more holistic approach, considering the possible connection between some not yet nonsense regions. Well, I'm not sure. So your definition of nonsense regions, so I'm not sure what you mean by that. But if you are referring to, so I mean, we don't have an opportunity to talk. So I'm not sure what you, but I mean, so for example, viruses typically, I mean, unlike eukaryotes, you have intergenic regions, you have genic regions. In this case, I was referring to proteins. So you have mutations, and we are talking about protein coding regions, mutations in the protein coding regions. So I'm not I'm not sure what you mean by nonsense regions here. So moving on, how did you decide Shannon entropy as a metric for diversity decisions? Okay, well, there are many metrics for diversity analysis, you could use different ways to study. What's nice about Shannon's entropy is that it captures the different sequences, the different distinct variants, and it also looks into the frequency of each one of them. And we were able to, of course, expand that to different chimers. And so we were able to, we found it robust in the sense we could, easily apply it. It accounted for the different sequences, accounted for probability. We could do different chimers, and we could also do something called, we could even do correction to that entropy, whereby because of sample size, your entropy may be not reflective of the true entropy. So imagine if you had a bigger sample size, the entropy might be a little different. So we could even do correction using Paninsky's rule to get much more representative entropy. So again, it's one of the ways you could study. I'm not saying it's the best way. There are other ways that people study diversity, but there are also many publications that have looked into entropy, although I think a number of them focus on just a chimer of one. So they just look at the one, each amino acid positions. In our case, we looked at monomer because we felt we were focusing on antigenic diversity. So that's our reason for focusing on a chimer. So yeah, I think there are many other tools that could be used for as a metric for diversity. Do you think one metric is a good way of measuring this? Of course not. One metric is not good, but we wanted to take one metric that was robust enough and then go deeper. I think the metric to us was good enough. Each of the metrics have their own pros and cons. Yeah, we could use different metrics to study I think that's highly recommended. We could add on and complement that with different metrics, but our goal was to use one that's reasonable and then as acceptable, and then we go further. And we wanted to move beyond from just looking at entropy. We wanted to do the diversity dynamics. So we just chose one and proceeded over there. But yeah, you could use different tools also. It wouldn't be better to use more metrics at the more confusing results, since they might have perfect consensus. Yes, you could like you're saying, you could do different methods and look for some agreement, consensus between them. Yes, they might still be useful despite. So that's actually a whole different area. One could actually look at diversity between, and one could even do a benchmark analysis of different diversity tools and really come up with one tool or recommend one tool that does very good overall. And I don't think there would be one perfect tool for everything, but make a benchmark analysis to do recommendations. So that's something that could be done. We have not done that, but that's an area one could explore. It's very nice that you mentioned these different method, Robinson, Rechavi, tissue specificity method. So I would look into that. But there are, I think this is a common problem in bioinformatics when you have different tools out there. Consensus, of course, is a good way to go. Maybe following question would be more, how do you confirm the significance of you discovered in silico? Okay. Significance, I think, so some of the take-home messages would be, for example, to me, one of the highlights was the unique variance. So the fact that we were able to see the unique variance behavior and identify the positions where this is occurring, and this is a real thing. It's really there. We have positions where you see sequences once and they disappear, and this is experimental data. Based on what's available. So the significance is, when we first saw this, we just couldn't believe. We actually thought this could be sequencing errors, but we saw them at almost every position, and there were some positions that were just so significant that they couldn't be sequencing errors. So this is like a behavior of the virus that we had not recognized previously. And so that's one significance of these motifs, that they show us some, what do you call this, subtle or hidden behaviors or patterns within the diversity of the virus that may not be obvious. There's a word I'm looking for, can't recall, but so yeah, that's if you're saying in terms of validating experimentally, I don't think, depending on what we are saying, the data we have is experimental data. This is sequencing data. So, and then the motifs are just describing, the patterns within the data. So, in terms of validation, I don't see what is there to validate, except that, you look at the patterns, and then based on the conclusions you make for that patterns, then those patterns, then you could come up with some validation. But for example, the index, if you go for those index positions that are highly conserved, if you make statements like they would be ideal, they would be good for vaccine design, well, they're highly conserved, so they're good candidates. But whether they really trigger an immune response, you could do prediction, you could do match with reported epitopes, but you still need to do some experimental validation to see if they actually do trigger an immune response, and they would be good for further exploration. So that's something that you would need to confirm the significance of the index as vaccine targets. But otherwise, they allow us, from entropy to zoom in into that entropy and to see what is really going on from the perspective of the different players that drive that diversity. And we learned a number of things from there, and as shared. And I think the more fascinating is this motif switch, which is also very complicated. But hopefully we'll also learn the significance of how one mutation can at times. So you can have a mutation, and there's you don't expect much change in the incidence, but imagine you have a mutation and it changes the motif from it being a minor to becoming a major. So that's a change. there's a change in terms of mutation, but that is accompanied with a jump in the frequency, so much so that a motif becomes from minor to major. And there are times we have seen, a minor can become even an index. So we are very interested to see and further understand how those actually happen, and what are the implications of those changes in terms of vaccine design, for example, or in terms of fitness selection of the virus to be a much more fit virus in maybe spreading more rapidly in the population. So those are some further questions that remain and remain to be answered and merit further investigation. All right. Okay. So might not have a perfect consensus, I think. Oh, okay. So
Dr. Khan, let's call this last question, because in 3 minutes the session will end automatically.
Okay, go for it.
Okay, you were speaking.
Oh no, I think I've answered all the questions. So
okay.
Unless they're, yeah.
Okay, then we would like to thank you for your contribution to our webinar project. If you haven't follow us on social media, you can use the links on the sticky message, or if you are watching this video on YouTube, we are going to share those links in the description below. So I would like to thank Dr. Khan on behalf of RSG-Türkiye and on behalf of our attendees. Thank you so much. And I think we can now say goodbye.
All right, my pleasure. Thank you everyone for joining. I hope it was useful. Thank you, Engin, for your hospitality.
Thanks so much.
Okay, cheers.
Thanks so much.
Bye-bye.
We would like to see you again.
Likewise, I hope to be able to contribute in the future.
Thanks so much.
Okay, bye-bye.